let's get started. So, I am Dakota Dywert, also known as Dakota 2. Today we're going to be talking about few eccentric exercise responsive genes relate to body mass index. But before we get started, let's talk a little bit about myself. I got my bachelor's in exercise science from Franklin College in 2020. Then I got my master's in exercise physiology at Ball State. And I'm currently a first-year PhD student in exercise science working with Dr. Hubel. And as of now, my current research interests are how does exercise-induced muscle damage and obesity relate to each other? So that said, let's get started. So as Dakota talked about, we know that eccentric exercise induces exercise-induced muscle damage, which then also can lead into inflammation, strength loss, and microstructural changes here, such as Z-line streaming. We also know that some of these interact with each other. However, as Dakota mentioned as well, we can also look at gene expressions, or what is the muscle thinking once you induce muscle damage. And we know that these markers have different gene expression profiles, or we can call them eccentric related genes. However, we also know there's genes related to BMI. So the question is, how do eccentric related genes and BMI related genes overlap? Or simply put, how is BMI changing what the muscle is thinking and causes any changes in gene expression? So very similar to Dakota's methods, we had 29 males do eccentric exercise, 10 sets of 10 on the Biodex. The average age was 20, the average BMI was 25.5, which is important in the discussion. 3 hours later, we had muscle biopsies done of the vastus lateralis, of the non-dominant leg. And then we got DNA microarray, which was assessed using an Agilent scanner, which then told us gene expressions such as how many genes were eccentric related, how many were BMI related, and how many were both. And that was assessed using a repeated measures ANOVA with a p-value of 0.01. So let's look at the results. So in terms of eccentric related genes, we saw there was 1,908 genes that responded to eccentric exercise, which is around 10% of our total genes. There's about 20,000 genes in the human body. So there's a very significant amount of genes being activated and turned on from eccentric exercise. In terms of BMI related genes, there are only 64 that were turned on. And then in terms of what was interacting or overlapping, there was only 3. So what does that mean? Well, there's less than a 1% overlap between eccentric and BMI related genes, suggesting that BMI is not really changing how the muscle is thinking in terms of turning on the eccentric related genes. However, there's a caveat. Our participants were healthy. They were relatively young, college-aged males, and they had a slightly elevated BMI. So if you look at this box plot, this kind of tells you where the individuals lined up for BMI. As you can see, there's quite a few that were lower on the end, but there were also a few that were at above that obese range, right? 30 is about the marker for obese BMI, but a lot of them set slightly above that elevated range, okay? So why is this important? Well, if we consider some other comorbidities, such as individuals who are actually obese, so if we considered individuals who are more in this range, we might actually be able to see a significant interaction between BMI-related genes and eccentric-related genes. Or what if we looked at sarcopenia, which is simply just age-related muscle loss of muscle. So if we looked at these kind of comorbidities, we might see that there's some changes in the eccentric-related genes. Again, why is this important? If we look at, it could have training implications. So if these individuals have exacerbated muscle damage just because they have sarcopenia or obesity, we're going to want to try to maybe start lighter so that they aren't experiencing those symptoms, or maybe consider pre-training before we actually put them through a resistance training program. So if there are any questions, there's my email, and here are my 2 cats. So again, let us know which ones are cuter, and thanks for coming to my TED Talk.